you very much, Jerry, for a wonderful talk. And but listening to your so-called technical talk today, I'm sure we all look forward to your two so-called popular talks. I'm sure it'll be fantastic, considering that how easy going these talk was. So, questions?
So in your talk, you mentioned that females have a tendency of avoiding mating with the males of different species.
Yes.
Because they have to invest a lot of energy in the entire process. On the other hand, the males do not have to invest so much energy. So if you look at the movement of males versus females in these zones, do you observe males to be more mobile as compared to females? Do they have more tendency of moving around?
Well, that's a good question. I mean, that was the first thing we thought of when we saw the male hybrids at the top, that they're more mobile. We tested that in the laboratory in two ways. Now, granted, these are not perfect tests. First of all, we put banana bait at one end of the building, and we released the flies at the other end, and we saw who came to the bait first. And they came in both equal numbers. Well, that could be just, detecting bananas. It doesn't necessarily mean mobility. So the other thing we did is we built this huge tower of PVC pipes, and we put flies at the bottom of it, and then we let them go. We didn't disturb them, and then we just observed their mobility vertically. I mean, this is the best we could do to make an artificial mountain in the laboratory. But again, there was no difference in mobility between males and females in either experiment. So if there was a difference in mobility, you have to explain, first of all, why it's only the one kind of F1 male that gets to the top. Why not the pure species if the males are more mobile? And not only is it just one F1 male, it's just one of the two F1 males. So you'd have to make the hypothesis that first of all, the hybrid males are more mobile than the pure species males, and second of all, that only one of the two types of hybrid males is more mobile. Now that's possible, but the tests we've done in the lab using all four genotypes, we have never seen that occur. So the short answer to your question is no, there doesn't seem to be a difference in mobility.
Your correct phylogeny suggests that there was a secondary reinvasion of Yakuba onto the island.
Yes.
It must have given some time for the species to evolve before the secondary reinvasion happened.
Yeah, probably about 400,000 years.
Okay, so can we estimate that time, which will then sort of give us an idea of how long it would take before it can tolerate coming back of this?
Yeah.
And secondly, is there any ecological or any other kind of evidence that there was not this reinvasion for those 400,000 years? I mean, why did it happen now? Why did it not happen all during that period? Is there any other independent evidence?
Yeah, those are good questions. The best we can guess, well, first of all, I mean, we have independent evidence that most speciation is allopatric in Drosophila. Eleanor and I have done some work on that, but it's not just in Drosophila. I mean, there's very little evidence for sympatric speciation again. So we think that probably this is a rare event, because it's hard for a fly to fly 250 kilometers over salt water. So just without knowing, I would guess, well, the second invasion there would have to be some time. There would have to be enough time for that first species to have evolved reproductive isolation before the second one came. Now, the best guess we can make is based on the data of the mitochondrial divergence. It would be about 14,000 years ago. And so if you posit that mitochondrial invasion happened when the species encountered each other, and 14,000 years ago, the island was all rainforest. There was no cutover area. So it was probably not suitable for Yakuba so much. And so probably even if it made it there, it wouldn't be viable. But if the mitochondrial invasion happened soon after the second colonization, then it would be about 14,000 years, which means that the speciation event occurred 400,000 years in the past. And then the second invasion happened just fairly recently within the human time span. We don't know that for sure, though. All we know is when the mitochondria invaded. It could have been, well after those species had been in contact with one another.
So this hybrid male at the top mystery that you mentioned, a piece of that, if you're just getting males from one set of crosses up there,
yes,
at the lower hybrid zone, you should have more females. Is that true?
No, we don't. Actually, at the lower hybrid zone, we find, first of all, we find males and females in roughly equal numbers as they're produced in the laboratory. And we find both kinds of crosses. Santamea by Yacuba and Yacuba by Santamea. So there's something peculiar about that upper hybrid zone that only attracts one kind of hybrid, and it's only one sex of one kind of hybrid. So, the lower hybrid zone is very different for some reason, as it should be. I mean, they behave normally there. You get mostly the right kind of hybrids, and you get them in 50-50 proportions. So,
another question I had about hybridization is that as we are looking at genomes more and more, we find signatures of hybridization usually way far back in the past in a lot of organisms, from bacteria all the way to what we call more complex organisms. But in Drosophila, you showed that at least in this species pair, there is not much hybridization going on.
Yeah,
if you
correct.
look across all species, how many of them readily made and how many of them have naturally exchanged genes? Is there any understanding?
Yeah, nobody's really done this in other species of Drosophila because, well, first of all, because they have to be sympatric. And most of the species that people are interested in are not sympatric, so there's not even any possibility of gene exchange. Nobody's really looked at that at all. I'm trying to see if, in areas where species are sympatric and they hybridize, I think I can only think of 2 or 3 cases where any of the hybrids are fertile. So there's no possibility of gene exchange, but there might have been in the past. So, the fact is that we don't have that kind of data for other species because most of the species are not possible to do this kind of work in. And now, of course, to look at hybridization properly, in areas of low recombination, you need a control pair of equal age and presumably in the same group, and we just don't get that in any other group of Drosophila. So yes, it is possible, but not many people, nobody's done it, in fact, except for us. I doubt, we can look at some things. We can look at chromosomal inversion polymorphisms, which are fixed between species. And we do not find sharing of that kind of polymorphism between species that can hybridize, like Drosophila pseudoobscura and Drosophila persimilis, very famous pair work done by Dubchansky. And they hybridize in nature, but we do not see inversions moving between the species. But those are inversions, so they have whole bunches of genes in them. And looking at individual loci would be much more informative. So.
Most of the textbook definitions of species attributed to Ernest Mayr that I've seen go something like this. 2 populations belong to the same species if and only if they have the potential to mate to produce fertile offspring.
Correct.
That doesn't seem to be the definition that you're using. In fact, the definition that you're using seems to be not a crispy set definition, but something that I would call either fuzzy set or prototype definition. What exactly is the definition of species that allows the possibility of hybridization? Because if you use the textbook definition, hybrid species is logically impossible.
Yes, correct.
So what is the definition then?
You're talking about what my definition of species is. I can be weaselly and say read chapter 1 of my book with Alan Orr. We do use a fuzzy definition because speciation is a continuous event, and there are some things that we would consider good species, like these, where there is some gene flow at all. So what I like to do is to use what I say, there's a sort of gradation of speciation. Ideally, 2 species that live in the same area and show no gene exchange, nobody argues that those are good species. What about if there's a little gene exchange? Well, I would call these different species, but there's a little gene exchange. So what I say in the book is that there are species that are more or less species-like, that conform more or less to the ideal Meyerian definition of species. But when, there's some fuzziness, well, you make a judgment call. Here I would say most people would call these different species. I think that what I can say in fairness is that almost everybody, regardless of whether they're systematists or have other species definitions, when they actually come to work on speciation itself, they study reproductive isolation. So somebody like Joel Craycraft, for example, at the American Museum, who doesn't adhere to the biological species definition, he uses a genealogical or a lineage definition. If people like that actually work on the process using real organisms, they always wind up studying reproductive isolating barriers, which to me says that there's at least intuitively involved in the idea of a species as something to do with reproductive isolation, whether or not it's complete. So that's sort of my Weasley definition. We can tell absolutely if two things are different species and good species, like us and chimpanzees. No gene exchange possible. By the way, that's been tried, at least in one direction, but that's in the book. I won't talk about that. Things where there's a tiny bit of gene exchange, and when it becomes populations instead of species, that's more or less a judgment call that, you know, nobody can actually, there's no species definition at all that will give you an absolutely clear definition of what a species is in all cases.
So I'm curious, does your definition allow a single population to belong to two species at the same time? In fact, this is what you would expect given ring species.
Well, first of all, there's no, you know, all the good examples of ring species haven't held up, and that includes Ensatina, the salamanders, the seagulls that are around the Arctic, and even the greenish warblers, which are the local ring species around the Tibetan Plateau. When you actually look at them, it's not a continuum of reproductive isolation. They all seem to have breaks that have occurred historically, and there's been allopatric species. And what you would call these things is anybody's guess. So even in the case of ring species, nobody, even Dave Wake, would say that different populations belong to two species. They were always called Ensatina, Schultzii, and then given subspecies names because there was supposedly continuous reproductive isolation. There was continuous gene flow along the ring species. But as I said, you know, the concept of ring species is no longer one that people can hold. The textbook examples have all shown to be erroneous in one respect or another, I think.
In fact, David Wake sort of distanced his own lifetime work of ring species.
Yeah, he actually admitted that he was wrong, but he didn't do it in a very explicit way. Because when you look at Ensatina, you find that it's not continuous, that there are breaks in the genes and the reproduction along the barriers, which implies, after all, this is 20 million years, and the Sierra Nevadas have only been up for about 10 or so, that the speciation was largely allopatric. It was not a period of a species spreading out and differentiating to the point where the ends were reproductively isolated. But there were barriers in between those species. So it doesn't correspond to the classic ring species definition.
You mentioned a couple of studies. I mean, you talked about speciation as well. My question would be this, that does proximity to the equator have any effect on the process of speciation, or is it independent?
Proximity to what?
To the equator.
Oh, well, what we know is that there are more species as you move towards the equator. Speciation seems to be higher, and there's a million different theories about that. One of them is that there are more biotic interactions at the equator than at the north. But of course, there are more biotic interactions because there are more species. So that's sort of a bit, you know, of a regress. Another one is that the environment is harsher in higher climes, and there are fewer niches for species to be in. But again, that's the problem of, you know, why is there fewer niches? Because some of the niches are biotic rather than physical niches. And so as far as I know, ecologists are still arguing about this. It's an indubitable fact that as you go from either the north or the south temperate zones towards the equator, you see a higher species diversity. The reason for that, as far as I know, because one of my friends is an expert on this, is not known. But I would say consult an ecologist or a biogeographer instead of me for the latest on that issue.
Yeah, I don't know it's relevant or not, but in viral variants, there's a concept called quasi-species. So do the hybrids fit into that kind of model?
Yeah, I'm not familiar what a quasi, maybe you could tell me what a quasi-species is?
It's a continuum of
Oh, okay.
different, because viruses evolve quite fast, so you're not looking at a single, when you look at a population, you're looking at n number of variants at a given point of time. So
Yes.
they term it as a quasi-species.
Yeah.
So
what you're talking about is a continuum of things, but yeah, sort of like a ring species, but yeah. Viruses and bacteria may correspond to those things. It's unclear whether they form these kinds of discrete clusters that organisms do. These hybrids would not fit that very well because the males are sterile. And one requirement of a species has to be that they're interbreeding individuals. So the males can't interbreed with the females, so it can't be a species. The females can breed with either of the males, but as you saw from the data, there's hardly any gene flow. And there's discrimination between the hybrids and the pure species. So I would say no, what we have is two good, pretty good species, not one perfect species, and that's it. And there's some gene exchange, but not very much between them. I mean, these things do, if you go out in nature, you don't really have much of a doubt of what you're looking at when you see a given fly. So, you know, that to me means that they're pretty good species.
So what about species which have sex determination independent of the heterochromatic chromosomes? For example, crocodiles and kind of stuff, where the temperature determines the sex. Do they follow this pattern of speciation?
Sorry, I can get the first part of your question?
So the species or
Yes.
which have the sex determination based on the, like say, temperature, which you know,
Oh, you mean like turtles
gene
and
specific,
stuff?
yeah, turtles
Yeah.
kind of thing, which it's not based on the two different chromosomes. Do they also fall into this kind of a pattern?
You're talking about Haldane's row? Well, if they don't have sex, if their temperature determined sex, I mean, you couldn't even test it in them because they don't have heterogametic versus homogametic sexes. So, I mean, you could say, well, are the males more likely to be sterile than the females, even though they don't have heterogametic sex? And that's a good question. And the answer is I don't know. I don't think that we have enough data on turtles to know that. What other organisms have temperature-dependent sex determination besides turtles?
Crocodiles. We have the same crocodiles, turtles. You know, these are
Yeah.
the ones.
I don't know. That would be an interesting thing to look at, and I don't know the answer to that. So.
Okay, if no more questions, thank you very much to Jerry Coyne.